Welcome to the second day of AquaSustain 2024. I am Anvesha Prakash, a master's student in the Department of Microbiology from Veterinary Science and Technology, and I shall be your main coordinator today. Today's session, or today's conference, will have the oral session preceded by the coffee time demonstration, technical sessions 3 and 4, and finally, the second big question workshop. Let us start with the first session for today, the oral session. Each presenter will have 15 minutes for their time, split into 10 minutes for presentation and 5 minutes for question and answers. At the 9-minute mark, the timekeeper will ring a bell as a remainder of 1 minute for your presentation, followed promptly by the Q&A. The moderator for this session is Dr. Seema Medhe from Chulalongkorn University, Thailand. Over to you, Doctor.
Good morning, everyone. I'm Dr. Seema Medhe. I will be the moderator for today's oral presentation. We have 6 oral presentations scheduled for today. 1 from natural productivity, 4 from One Health approach, and 1 from climate and climatic events. Each presenter will be given 10 minutes for presentation, followed by 5 minutes for question and answer. I request every presenter to finish their presentation within allotted time. So our first presenter is from the One Health approach session is Mr. Amrendra Kumar from College of Fisheries, Dr. Balasaheb Chavan, Konkan Agriculture University, Dapoli, Maharashtra, India. His presentation title is Antibiotic Resistance in Pseudomonas aeruginosa, a critical challenge in Nile tilapia aquaculture. Mr. Kumar, please start your presentation.
Very good morning to everyone present here and virtually attending participants. I myself, Amrendra Kumar, I graduated recently from Ratnagiri, India College of Fisheries. This is my first international presentation, so you can understand what's going inside. So my topic is on antibiotic resistant and their mechanism in Nile tilapia. And the bacteria is Pseudomonas aeruginosa. We all know the tilapia is most third, one of the important cultured species in the world, including Asia and Africa. And it is a native species to Africa, and it has been cultured in Asia and other countries in very large quantities. So there are always a large number of microbials which are already in our ponds. So to manage them, farmers always use antibiotics and without any prescriptions sometimes. And this leads to create antibiotic resistant strains in aquaculture system. And recently, from WHO has published a list to which bacteria the new resistant antibiotics are urgently needed, and this bacteria comes under that list. So you can understand the critical situation of this bacteria, antibiotic resistant in this bacteria. So what we did, first we searched for the bacteria which is really a concern in right time. So we got to know that this Pseudomonas aeruginosa is one of them, which really needs attention of ours. And so after that we started to search the papers available online, and we got to know that there is plenty of papers available, around 2,660, but we restricted our review to only tilapia. So we got finally 24 paper which we reviewed in our paper. So we, okay, sorry. And in result, we noticed that this bacteria has reported in Egypt, Uganda, Kenya, Tanzania, Vietnam, India, and China. And not only these countries, apart from these countries, in Thailand also, in other countries also, in this bacteria has been reported. But due to some restrictions, we had not included that paper in our review. So you can see, I have made a table which shows that major antibiotics, then the antibiotics under that major antibiotics, and their mechanism through which this bacteria is resistant to antibiotics. And I have put the reference. So when we were studying about the antibiotics, which are among them, so we got to know that beta-lactams and penicillins, these two were the most used and most commonly used antibiotics by the farmers. And all these studies were conducted on the ponds, not in like the fish were bought from the pond and they were analyzed in the laboratory. So we got all these results are in vivo. So we can understand the farmers at which extent they are using antibiotics. And apart from these two, aminoglycerides and then macrolides, quinols, tetracycline, all these are also a point of concern. So this picture illustrates the antibiotic resistance mechanism. So we all total found 7 mechanisms through which this bacteria is resistant to antibiotics. First one is, we all know that efflux protein. What happens? The bacteria efflux the antibiotics, whatever you applied in the system. So it is of no use to use these antibiotics in aquaculture system. Like that also, like that only, enzyme deactivation, then antibiotics alteration, then new protein formation against the antibiotics. It's all evolving with the time and with the extent of use. And in a conclusion, we get to know that this bacteria isolated from Nile tilapia is resistant to wide number of antibiotics. So instead of going searching for the new antibiotics to kill this bacteria, I recommend that we must approach to the novel process, novel solution, like vaccines, vaccinations, and other herbal products to combat with this bacteria. Alternative treatment like treatment with mixing of more than one approach like vaccination, and with that a little amount of herbal products and all these medication we can use. And better to eliminate all these resistant bacteria from the aquaculture. It's better to eliminate them and use biosecurity to avoid the contamination to the pond. And for further in detail review, it is recommended that systematic review and meta-analysis could be helpful to analyze in detail so what's going on, because we have seen in the second slide. So there is more than 2,000 paper available online. So it is really needed to analyze, assess these all data in order to get the proper solution for the future for the sustainable aquaculture. Thank you. So you can ask any questions.
Thank you for your wonderful presentation, Amrendra. Anyone have questions?
Very much. A very good presentation. Well done.
Thank you.
Your first conference, and congratulations.
Thank you.
And a very interesting subject as well. I mean, Pseudomonas aeruginosa, we normally think of it more as a human pathogen. So one thing I would question when you did your review is just be careful the methodologies that they'll be using to identify these organisms. And I'm sure you're doing a lot already with your professor, but make sure that you've got that the papers that you're reviewing have been using the recommended methods for identifying the pathogen. That would be one little thing just to make sure, because in the literature, there's quite a lot of, maybe not with the organism, but some of the organisms, when you look closely, maybe the identifications are not as robust as they might be. which can affect the validity of things. But I'm sure that's fine. It's just something to bear in mind. But thank you.
Thank you, sir. Thank you for your tip. Thank you.
Thank you, sir, for your valuable guidance. Thank you, Amrendra.
Thank you,
Let's give a
everyone.
round of applause to
Thank you, everyone. Thank you.
Our second presenter from One Health Approach session is Miss Andrea Pardo from Aquaculture and Aquatic Resources Management, School of Environment, Resources and Development, Asian Institutes of Technology, Pathumthani, Thailand. Her presentation is titled Vaccination of Nile Tilapia, Broodstock with a Bivalent Vaccine against Aeromonas veronii and Streptococcus agalactiae. Ms. Pardo. Ms. Andrea Pardo. She's not in the hall. What to do? Ms. Andrea, please start your presentation.
I apologize. Okay, good morning, everyone. My name is Andrea, and I'm a PhD candidate at the Asian Institute of Technology. So today I'm presenting our research on vaccination of Nile tilapia, broodstock, with a bivalent vaccine against Aeromonas veronii and Streptococcus agalactiae. Tilapia. Tilapia is an important fish in aquaculture, and actually this is one of the most farmed aquaculture species in more than 140 countries worldwide. It is a high nutritional value species and grows fast and actually adapts very well in different environments. However, current bacterial diseases are a big problems for farmers. I apologize again. Okay, so let's continue here. Streptococcus agalactiae and Aeromonas veronii are the main causative agents of two of the current bacterial diseases of significant importance in tilapia aquaculture. The one is Streptococcosis, which is actually the most frequently reported bacterial disease in tilapia aquaculture, and the second, Aeromoniasis. So fish that are infected with the Streptococcus agalactiae will usually present clinical signs such as erratic swimming, exophthalmia or pop eyes, and high mortality rates, while fish infected with Aeromonas veronii might present symptoms such as lethargy and loss of appetite, skin ulcers, and enteritis. So why Aeromonas veronii and not Aeromonas hydrophila? Okay, while Aeromonas hydrophila has been traditionally considered one major pathogen in tilapia aquaculture, in previous studies it has been found that Aeromonas veronii was the primary pathogen in infections in tilapia farming, which provoked severe disease and high mortality rates. So this is why trying to combine the vaccine, which contains Aeromonas veronii and Streptococcus agalactiae, might provide a wider range of protection. So the objectives of our research was to investigate the human immune kinetics of Nile tilapia broodstock post-immunization with an inactivated bivalent Aeromonas veronii and Streptococcus agalactiae vaccine. Second, to assess the maternal antibody transfer from vaccinated broodstock to the offspring. And third, to assess the protective efficacy of this vaccine through a passive immunization assay. So for the preparation of the vaccine, of the bivalent vaccine, what we did was to culture the isolates of Aeromonas veronii and Streptococcus agalactiae in TSV, and then heat inactivating them in water. To confirm inactivation, we had to spread in TSA plates, and after no growth was present, then we could confirm the inactivation. So after that, no further processing was done. We used this bacterium as the vaccine. And after inactivation, what we did was to optimize the concentration of both bacteria to have the final formulation. After that, to start with the experiment, what we did was to inject 200 microliters of these vaccines to the fish and provide injection boosters on day 28 and day 23. Blood, mucus, and egg samples were collected every week, so later we could assess IgM antibody levels using direct ELISA. For the passive immunization experimental design, what we did is actually the fish collected from the broodstock, we grouped them to have clean fish, and then when they reach around 20 grams, what we did was to inject 100 microliters of the sera from the vaccinated broodstock, and then challenge them with these two pathogens, Aeromonas veronii and Streptococcus agalactiae. So, as results, in the broodstock vaccination, after primary vaccination, we can see an increased levels of IgMs in the vaccinated fish compared with the control groups for both bacteria. And after the boosters on day 28 and day 63, this significant increase was seen of these IgMs, again for both bacteria, but during all the study period, there was a higher IgM levels for anti-Aeromonas veronii compared with the Streptococcus agalactiae. So after the booster, what we see is that there was a strong secondary response, and it can be seen clearly with the Streptococcus agalactiae. In the results for the assessment of maternal immunity, we observed that there was a transfer of IgM antibodies in the fertilized eggs. However, these antibodies were not detected in the fry after yolk sac depletion. So this might indicate that there is a short persistence of passive immunization, and actually this finding is similar to the ones found by Dr. Tao on TILV, that actually they found that there was no detection of antibodies after 1 or 3 days post-hatching of the eggs. In the passive immunization, the fish that were vaccinated with the sera from the broodstock, they actually present higher survival rates compared with the control groups, which indicates that this sera coming from the vaccinated broodstock indeed can protect against infections of Streptococcus agalactiae and Aeromonas veronii. And of course here we did not measure other components such as antimicrobial peptides or complement proteins, but we only did with antibody IgM. However, when we inject the sera, what we did was to inactivate by heating it at 40 degrees Celsius for 20 minutes, so we can ensure that we are focusing on the IgM antibodies. So, as conclusion, this research provides insights into the dynamics of the humoral immune response in Nile tilapia broodstock with higher levels of anti-A veronii antibodies compared with anti-Streptococcus agalactiae antibodies following primary and booster injections in vaccinated fish. This study also demonstrates the evidence of maternal immunity transfer from broodstock and fertilized eggs following the immunization with the bivalent vaccine. No specific antibodies can be detected in the fry after yolk sac depletion, indicating a short persistence of this maternal passive immunity. This approach holds a promise for vaccinating broodstock as a biosecurity measure to reduce the risk of vertical transmission, therefore providing support for the sustainable of aquaculture, of tilapia aquaculture. So further studies required to demonstrate effective broodstock vaccination in field application. So I would like to give my thank to the AIT scholarship from the Asian Institute of Technology, and to my mentor, Dr. Ha, for giving me the opportunity to get the student assistantships from the Fagos of Animal Health Projects, and to Pisantri, Pilompan, and Pipran for helping me when I got sick with anemia. So thank you very much.
Thank you for your wonderful presentation, Ms. Pardo. Do anyone have question?
Excuse me.
The quantity of antigen given to the fish, Were they the same or different?
No, there were actually, we had a previous small experiment in which we test different formulations. So the formulation number 4 is the one that we chose, and actually it has higher levels of antigen for Streptococcus agalactiae in lower levels for Aeromonas.
Okay, thank you.
Another question, please?
So, anyone have question? Thank you. Let's give a round of applause to Andrea.
Thank you.
Our next presenter is from One Health Approach session. Is Mr. Vin from Aquaculture and Aquatic Resources Management, School of Environment, Resources and Development, Asian Institutes of Technology, Pathum Thani, Thailand. His presentation is titled, Do Autogenous Bivalent Streptococcus agalactiae, Serotype 1 and 3 Effectively Mitigate Streptococcosis in Red Tilapia? Mr. Vin, please start your presentation.
Good morning, everyone. My name is Vin from IIT. Today, I'm quite happy to be here to share with you my research I have conducted recently. I tried the autogenous bivalent vaccine to prevent streptococcosis in tilapia in the farm. As you know that tilapia is an important species in aquaculture, and is considered a candidate of sustainable aquaculture because they grow fast. They are not picky feeders, and they can feed on many things. So they tackle nutrition problem and food security. And for some countries, especially for low to mid-income economy, they are quite important for their social economy. However, like in many other species of aquaculture, they are infected with many diseases that may prevent the further expansion of tilapia in the world, especially the streptococcosis. The main culprit is the Streptococcus agalactiae. And currently, based on the capsular polysaccharides, we have identified at least 10 serotypes, 1A, 1B, 2 to 9. And in Thailand and many Asian countries, serotype 1A and 3 are commonly encountered, and they can cause significant mortality up to 70%. And for some clinical signs, when the fish are infected with Streptococcus agalactiae, they may show eye damage, skin hemorrhage, slow swimming pattern, and internal hemorrhage and fluid accumulation. To prevent the disease, vaccination is considered a sustainable, non-antibiotic measure. As you know, that the vaccine prepared the fish with a specific immunity. So for the next time they got infection, they will have a better way to combat the disease. So because of this reason, many studies have been focused on Streptococcus agalactiae vaccine for tilapia. However, many research usually focus on only one serotype of this bacteria. And in the market, we have many commercial vaccines for them as well. For example, vaccine from Pharmacure, MSD Animal Health, Worldbug, SA recently. However, Streptococcus agalactiae can be quite sneaky because when we apply the vaccine, we need a specific serotype to cover the protection, and it will be less effective because of the coexistence of many serotypes. And even so, when we try with multiple valent vaccine, because they are quite diverse in the strains, it will render the vaccine will be less effective. So we are thinking that what if we can apply local strain to them to enhance protection? And that actually matches the definition of autogenous vaccines. In short, autogenous vaccines employ local strains isolation, and when we recover the isolation, we produce a vaccine mostly by inactivation method, and we apply to the same area. So in aquaculture, they have shown to reduce antibiotic use and AMR. So our objective is that we produce an autogenous bivalent vaccine to prevent streptococcosis in the farm. To get the vaccine, first we isolated the bacteria serotype 1A and 3 from a farm from Prachinburi, Thailand. And then we upscale the bacteria, and to provide the vaccine, we heat kill them at 60 degrees for 1 hour. And then we mix both serotypes in equal concentration for the bivalent vaccine. For the trial, we got lab-scale experiment and a farm trial. We injected them with 2 doses, a primary dose at day 0 and a booster dose at day 28. And for the lab trial, we monitor IgM antibody every week. And in the end, we challenged them with 2 models, an immersion challenge for 5 weeks and an injection challenge with separate serotypes for 2 weeks. For the farm trial, we also monitor mortality, cover the crop from February to July, and we also monitor antibody of the tilapia every 3 weeks. And here's the farm trial. We put them to half-hour units. Each half-hour consisted of 300 fish. Each treatment consists of 4 half-hours, representing 4 replicates. And this is the real picture of the farm. To compare the antibody level between control and vaccinated group, we use the Wilcoxon test with a cutoff level of 0.05 significant level. And for survival analysis, we use a log-rank test. So I will share some results. First, we got the antibody response IgM of the fish in the lab. As you can see here, the fish responded very well to the vaccine, and the significant level of IgM started to grow since day 7, but at quite limited level. But after the booster, you can see at day 35, they grow significantly. And even after the immersion challenge, they further grow after only 3 days. And for mucosal response, it's much less than the IgM seen in the serum of the fish. And in the end, with the challenge experiment, we found that when the control fish was injected with the bacteria, they die almost 100% after 14 days. Meanwhile, the vaccinated fish survived very well. Again, serotype 1A, relative percent survival was around 97%, and that 86% against serotype 3. So this is the result of antibody from the farm. And you can see that after the booster, high antibody level were observed over the crop for 4 months. And serotype 3, the response was less than that, and it appears to diminish after 84 days. And in the end, after the farm trial, we found that vaccinated fish survived very well at 94%. Meanwhile, the control only survived 66%. So that the vaccine helped to enhance the survival rate by 41%. And that also increased the productivity by 45%. From our experiment, the autogenous bivalent vaccine actually stimulated really high degree of adaptive immunity in tilapia. And the first booster served as a Brahmin dose, so they will trigger a low degree of antibody response. Follow that, the booster was required to boost the immune response further, and it is more potent. In the mucosal response, significant level of IgM only see significantly after the booster, and the mucosal the IgM expression was so large comparing to the systemic. And we tried with the immersion challenge. It's quite different from many other research because we want to mimic the natural infection via the fish epithelia. And you can see after only 3 days, vaccinated fish rise the antibody very high. Meanwhile, among the control fish, very low level. So it may present a challenge when we try with the immersion vaccine in aquaculture. And for the farm trial, because the antibody cover over the crop, so it can protect the fish very well for the whole crop. And you can see that there are some fish got the infection because the IgM rose higher than the cutoff here. And the response against serotype 3 can be quicker than serotype 1A because it was less persistent, and the peak of IgM levels are also lower in serotype 3. And in the farm trial, only over 80% of fish have insignificant IgM level after day 84. And the capsular structure of serotype 3 can be less immunogenic as seen in human. So that may explain the result from here. And the vaccine improved the survival by 41%. And in the lab trial, there was no significant difference between the survival against serotype 1A and 3. Although you see the level of IgM from serotype 3 may be less significant than serotype 1. And in further research, I think that cell-mediated immunity may be important that if we want to capture a full picture of the fish immunity. And for a farm trial, it's important to replicate, to validate the consistency of the vaccine. And multiple infection in the farm can be quite challenging to address. So some fish may die not because of Streptococcus agalactiae. In conclusion, our vaccine is very highly effective against the pathogen, and I think it can be applied in the farms and contribute greatly to sustainable aquaculture for tilapia farming. I would like to thank IIT and IDRC for the sponsorship with my scholarship. And this study was initiated and funded by the IDRC Canada to reduce the antibiotic use and AMR in aquaculture, and Chantec stream for bacterial isolation and serotyping. I also thank NAMSAI Farm for the agreement to test our vaccine. Thank you. So, do you have any questions?
For this session. Thank you, Mr. Vinh. Anyone have a question? So, please give a round of applause to our Mr. Vinh. We'll move towards the next presenter. Our next presenter is from the Climate and Climatic Event session, Mr. Viraj Daunde from College of Fisheries, Dr. Balasaheb Chavan, Konkan Agriculture University, Maharashtra, India. His presentation is titled Impact of Higher Temperatures on Mud Crab Aquaculture, a mini review. Mr. Daunde, please begin your presentation.
Okay. Very good morning, all of you. My name is Viraj Daunde, and I am from College of Fisheries, Ratnagiri, which is in India. So this is also my first international conference presentation, and from 5 August we are going to join CU WET for master's degree. So my topic is I have made a mini review on how higher temperatures are impacting mud crab. So the introduction, we all know that aquaculture is the fastest growing sector in the whole world. Aquaculture is going to be the future of our world, and it is feeding us in a whole different level. And if we come to the crustacean species, it holds one of the important space in the aquaculture, and the production in the 2022 of crustacean species were 12.75 million tons. And climate change, yeah, it's a very big problem in nowadays. It's a potential threat to the aquaculture production also. Nowadays also, the effects of climate change, these are getting visible. We are seeing in the summer, rain is there. In winter, some sunny days are there. Means more hotter days are getting. So the effects are getting visible. One of the effect is increased temperature due to climate change. It is having a very adverse effect on the aquaculture. Many species, their metabolism, their life cycles are changing because of increased temperature. You can see on in the picture, this is the basic pond aquaculture, pond farm of a mud crab. And you can see the temperature. If you compare in past decades, the temperature has, in the past 4 to 5 years, temperature has increased around 1.5 to 3 degrees Celsius if we compare the past decades. So why I have chosen mud crab species? It is a key commercial crustacean species in the Southeast Asia, and it has it is also located in Indo-Pacific region. It shows a good growth rate. If you compare the shrimp grows around 2 to 3 gram per week. And if you see at the mud crab, it grows around 10 gram per week. So it's a good growth rate. Plus, it is well suited for live marketing. Yeah, in Thailand, live marketing is very famous. And India also, we are improving live marketing. And mud crab is a very good and very easy to get to the market in a life. Also, crab fattening is very easy and a profitable business. Now, if we see how I have done this mini review, so I have taken around 25 to 30 research papers, and I have selected a specific results. These are the searches, means keywords I have used during my mini review, and I have filtered them. Mostly the criteria was the temperature should be above 30 degrees Celsius. So I have made a table which contains 9 research papers. I have taken 3 species of Scylla, and you can see I have highlighted some results which I have interconnected: O2 concentration, survival, feed intake, molting. So these are the common results I have observed during my review. So I have taken all these results and I have connected them. So I have made one diagram. You can see the higher temperature, due to temperature rise, water temperature is also increasing. And as an effect of that, oxygen in the water is decreasing. And as an effect of that, any organism in this kind of hypoxic condition, they spend most of their energy on survival rather than feed intake. That's why their feed intake of crabs are also decreasing, and it is leading them to the smaller size. It means their growth is also decreasing, and ultimately, due to low feed intake, they are moving towards the high mortality. The second thing is, when the temperature increases in water, the ecdysone hormone, which helps mud crab for a molting process, means instead of their molting frequencies are increasing, the crab which normally molts at 15 days, 20 days, they are molting on 8th day, 7th day, and after molting they get very vulnerable to diseases or cannibalism. So ultimately it is also leading towards mortality itself. Yeah, the conclusion that I've got is there is a positive relation between higher temperature and growth, feed intake, molting, oxygen, and mortality of crabs. And yes, higher temperatures are having very detrimental effects on the mud crab aquaculture, as we have seen. I have also recommended some points which is as a view of aquaculture. So to avoid this, we can put a very low stocking density in the pond for mud crab, because we have seen due to temperature rise, dissolved oxygen is getting very low. So if we keep the stocking density low, it will help them to survive more. The second thing is, if we want to avoid the direct contact of sunlight, we can put one green net above the ponds. Yeah, these are the references I have used during my review. And yes, further systematic review and meta-analysis is required to get the exact impact of higher temperatures. Yes, thank you.
Thank you, Mr. Viraj. Any question from audience?
Very good attempt as initiate on your part. Our institute was part of this climate change research in India, that is at ICR for more than a decade. I was also part of that. So there that agreed methodology was to correlate anything with climate change. At least we should have trained for at least 30 years of data. Please, just this is a suggestion.
Okay, yes, thank you. I'd like some suggestions also, because all experts are here from the globe.
So anything from audience, suggestions for Viraj and his study?
Maybe the cost for the farmer needs to be kept in consideration.
Okay.
The net, it's expensive.
Yeah, okay.
Thank you so much, sir. Thank you, Viraj.
Thank you.
Let's give a round of applause to Mr. Viraj. We'll move towards next presentation. Our next presenter is from the One Health Approach session, Dr. Pakkakul Sangsuriya from National Center of Genetic Engineering and Biotechnology, National Science and Technology Development Agency, Pathum Thani, Thailand. Her presentation title is Establishment of Continuous Cell Lines from Asian sea bass for the Study of Scale Drop Disease Virus. Dr. Sangsuriya, please begin your presentation.
Good morning, everyone. Today, I would like to present about fish cell line establishment and what is the application of the cell line in a fish disease. And the case study of the virus that we use to test is scale drop disease virus, or SCDV, in Asian sea bass. First of all, I would like to introduce our research team. Dr. Saengchan and me from Biotech, NASDA, and Professor Thanh Dong from AIT, and Professor Hirono, Professor Kondo, and Professor Sano from Tamsa, Japan. My talk is start with a scaled-up disease virus in Asian sea bass, the application of the fish cell culture, and the primary cell culture, and the establishment of the Asian sea bass cell line, and its characterization, and the final is the test of the cell line susceptibility to the virus. The scaled-up disease caused by scaled-up disease virus is one of the significant viral disease in Asian sea bass. In Thailand, the natural outbreak can cause mortality up to 40%. And the clinical sign of the disease is seen by scale drop, fin lot, and hemorrhage. You can see the particle of the virus in the infected tissue. And this virus is belong to the novel megalocytic virus in the family of Iridobilidae. And this virus found in Thailand in 2019. And the SDDV Thai strain shares high similarity to the Singapore SDDV is about 99.9%. And to understand more in this virus, we need biological tool to characterize or study of this virus in order to find a way to control this virus. And fish cell line is the answer for this. So I would like to talk about a fish cell line application. It is a powerful biological tool to study in many fields such as virology, toxicology, immunology, and genetics and pharmacology. For the fish virology, the cell line can be used to detect the virus and also characterization of the virus, also study of the whole range of the virus. And for the disease management, it is very important to produce the vaccine, especially for the virus vaccines. And this cell line can be used for screening of the drug or bioactive compound that's against the viral disease. However, at the time that we found the SDDV in Thailand, we don't have a cell line, especially the Asian sea bass cell line, which is the host of the SDDV. That's why we have to establish the cell line in our lab in order to study this virus. We use the explant technique to isolate the primary cell from the fish tissue, including head, kidney, brain, spleen, and fin. You can see cells. They migrate from the fish tissue and grow around the fish tissue. And these primary cells were subcultured for many times in order to establish the cell line or immortal cell. And finally, we can establish the fish cell line from the head, kidney, tissue, and we name it as HK. And this cell line can grow for many times. They can subculture for more than 120 passages. And the morphology of this cell is fibroblast-like. To prove this cell line is originate from the Asian sea bass, we have to confirm this cell line. We amplify the CO1 gene from the HK cell and then compare the sequence of this HK cell with the database. And the results show that the CO1 gene from the HK cell show high similarity, about 99.85% to Asian sea bass mitochondrial DNA in the database. So it can confirm that our cell line is truly from Asian sea bass, not from the other contaminated resources. Next, we have to test this cell line can be used for protein expression. By this experiment, we transfect the recombinant plasmid to the Asian sea bass cell line and see the protein expression. You can see in the picture, they can use for express the protein, green fluorescent protein from the recombinant protein. And it can conclude that this cell line is suitable for the study of the genetic manipulation. You can use this cell line for study the DNA vaccine expression to prove your vaccine is can express the antigen or not. Next, the important part of this study, we have to test these cell line can susceptible to SDDV infection. In this experiment, we use the scale-up disease virus isolated from Asian sea bat in Thailand to infect the HK cells. We used 2 doses, 10 to the 7 and 10 to the 8 copy. And these cells very sensitive to the virus. You can see that in the at first day post-infection, the morphology of the cell changed rapidly from fibroblast to a rounded cell here. When the infection progresses, more rounded cell can be observed. And finally, these cells detach from the flask and they die. And at 4 days post-infection, we determine the amount of the virus, and it can produce the virus around 2 to 3 multiplied 10 power 8 copy per milliliter. We also test that the cell line from early passage and the late passage, they still can propagate the SDD virus or not. And the results show that both early and late passage of the HK cell can effectively propagate the SDDV virus. And they show the amount of the virus around 2 to 6 multiplied 10 to the 8 copy per milliliter. So it means that we can use this cell line to produce the SDD virus for vaccine production. We also tested this SDDV propagated in the HK cell can cause pathogenicity in the Asian sea bass. We propagate the SDDV in the fish cell line and then correct the SDDV and then inject it back to the Asian sea bass with the different doses. For example, we inject about 10 to the 6th, 10 to the 5th, and 10 to the 4th copy per fish, and they show 100%, 90%, and 80% mortality respectively within 16 days after infection. And when we observe the tissue of infected fish, they show high copy number of the SDDV virus around 10 to the 5 and 10 to the 6 copy per 200 nanogram DNA. It means that the SDDV propagate in the fish cell line can cause the disease in the fish. And the clinical signs of the fish show the scale loss here and salary of fin lot. And they show the gill pellet and liver blood loss. And for the histology, they show the signs of a viral infection, including karyoraxis and pycnosis. And not only SDDV, we also test for the other virus in the Asian sea bat, including novel necrosis virus or NNV. And the results show that these HK cell can susceptible to the NNV infection. But they show a prolonged infection. For example, 8 days post-infection, they show clearly CPE. And they can produce the virus around 4 multiplied 10 to the 8 copy per milliliter. We also test other virus, ISKNV, infection spleen kidney necrosis virus, one of the important virus in the Asian sea bass. And it also show that these HK cells can use to isolate the ISKNV from the infected fish. So in summary of this study, the primary cell of head, kidney, brain, spleen, and fin tissue were successfully isolated. And the novel HK cell line can establish in this study. The origin of the cell line was confirmed by sequencing of the CO1 gene. And this cell line can use for testing the DNA vaccine expression. And the cell line can susceptible to the SDDV, NNV, and ISKNV infection. So the HK cell provide the power to powerful to study the viral disease in fish, and can provide more understanding of the virus. So if we understand the virus, we can find a way to control them, or we can use this cell line to produce the vaccines. So it enhance the sustainability of the aquaculture. So we would like to thank the funder from Thailand Research Fund, Biotech and Startup. And thank you all of you for attention.
Thank you, Dr. Sangsuriya, for your very innovative and interesting presentation. Any question from audience? Thank you, Dr. Sangsuriya. We'll move towards our next presenter. Our next presenter is from Natural Productivity Session, Dr. Basanta Kumar Das from the ICAR, Central Inland Fisheries Research Institute, Barakpur, India. His presentation is titled Integrating Pen Culture with Floodplain Wetland Fisheries, Enhancing Sustainability, Profitability, and Climate Resilience. Mr. Das, please begin your presentation.
Good morning to all. I am at the outset thankful to the organizer to giving me opportunity to give one of the topic of interest today, because all of you know wetland in India is sprawling over huge hectares, maybe 3.51 million hectares, in which community-based, culture-based fisheries are being practiced. So if the community-based fisheries are being practiced, and if you look into the things where stocking is culture-based fishery, the stocking from the outside to this, which carries a long distance, there is a mortality is there. So if you are looking into the animal welfare, less mortality, this is a technology where you can have in situ rearing, where you can grow the size, stockable size in the wetland, where maybe 4 gram fingerlings stock period of 2 to 3 months in a different maybe we are going for 3 lakhs per hectare. And that is a enclosure systems, and a pen is designed is 0.1 hectare. And in that, if you will stock that thing, and you will find this is a structure of pen. If you will find noticed here, it is a just 0.1 hectare. Maybe you form is a rectangular or circular type, and can be movable to the any depth. Height is based on the flooding conditions, can be raised from 2.5 meter to 3 meter, and where you can stock and have a feeding as per the desired level, and you can grow to a stockable size, because if you know wetland is rich in biodiversities, and there when a small size fish when stocked, there is a lot of cannibalism happens between the prey-predator relationships. So here you can, when you are attending minimum 20 grams and above, you can release into the wetland, and it will give a good amount potentials of the productions, and it will, when the community of maybe 100 households dependent in each of the things of a wetland in a cooperative mode, PFCA primary cooperative societies, it will add the value to their food basket. So if you look into these conditions, and we have done in the co-managements involving the society in the all aspect, and further that can be utilized, convincing the things, adapting the new technologies, and we have demonstrated maybe 7 to 8 states of India, starting from Manipur, Assam, West Bengal, Odisha, Bihar, and Kerala also, where we have adopted also integrated multi-trophic practices, where you have, if you see, Atrophilus suratensis, and your clams are being cultured in these cases. And if you see, these are some of these models of the culture-based fisheries. If you see here, some wetlands is rich in grass carps, where we have utilized the macrophyte controls with the grass carp models with the different stocking density, and it was found useful when these grass carps, after 2.5 months released to the wetlands, it has maybe more than 2 kilos within 6 to 8 months. That indicates without any systems you can increase the productivity of this, and with the different economics. And if you see Then we have how it is, if you see how it is increased in the wetland productivity from the different years, and that is a good practice if you look into this. These interventions, it has have a good productions in the wetland to which has increased up to the 32% also. So this is another way if you see. Again, we have taken into some of the major and minor carps, because when you talk about some of the climate resilient species in these systems, so we Indian major carps, rohu, katla, mrigal, we have a different stocking density we have practiced, and along with the labio bata also we take in these systems, and we found this is very well established, and again it has grown, like if you see productions of, if you see 682 kg per 0.1 hectare within the 90 days. Again with the, I am saying the grass carps also, there is 413 kg, which is a significant way of producing. It is another wetlands in the West Bengals. It is maybe transboundary nearer to the Bangladesh border. And if you look into this is the different, if you see, experimental harvesting data, and with its high BC ratios. Again, we do an Systemos sarana, which is an auto-recruitable species, which can breed itself in the things without going, because carps, they do not sometimes breed, and increasing the productivity and productions. So we tried with the Systemos sarana into these systems, and we find within 3 months, the result was very encouraging. And this is the, if you see, the maybe 748 kg we got in this, and that is also another wetland, 640 kg, so variations. So maybe if we are finding 6 to 7 ton per hectare production, so that will be effectively utilize the stocking practice and recruitable systems on the wetland. Again, if you look to these are the some of these, if you see the BC ratios and weight of the different stockable size, that made the farmers and cooperative society to come forward in a sustainable manner with these things. And where we have taken the different ways and productions, if you see, I told it's a recruitable and low yielding SIF species and high price demands. It will have a local preferred piece and a more price compared to the carps also. So it will fetch a good economic returns to the primary society people and the households also. And this is totally the co-management. Again, if you see, in the Assam wetlands, we have taken in a lot of wetlands in a study, which is starting from maybe from Bodo right to Lower Assam and Upper Assams. And with the everywhere, if you see, maybe Bata, Mrigal, Rohu, Katla, then some of the same combinations we have tried in the different places, and it was found a very encouraging results that leads to the practice into the farmers or the society people for wetland productivity enhancement. Again, you know, there are very less productivity, maybe the potential maybe 1500 kg per hectare in wetlands, but presently if in Assam you find maybe open wetland equation, every productions goes to 250 to 500 kg. So these interventions when in situ rearing systems, again the productivity can increase maybe from 1,000 to 15 and more also. Again, we generally stock in the culture-based fisheries 2,000 fingerlings per hectare and 3,000 in case of wetland. But when it is a smaller size, the recommendation size is 100 millimeters or 10 centimeters. But generally, the availability is this much size is very less. So people or the department used to stock less than, and that may be less than 10 centimeters, 5 to 6 centimeters. There is a more type of predations and cannibalisms. So this will help for preventing such type of mortality, and there is a reducing the populations in the environment, and it will increase the things. Again, today we are talking about the, again we have, again this experiment is also labiobata. Maybe one of these like your bata, then your mola. These are some of the climate resilient species, Pabda, which is a high value species in India. They have been tried in the different combinations. And we found also in a fen also there is auto breeding takes place by these species, which can be helpful for either increasing the productivity and productions in the wetlands also. And these are some of the experiments. If you see, the BCR is very high, and it will be found that that can be used for the practices in the wetland. Again, I told again some of the cartilage is also used individually in this, and you will find this cartilage is also very good for the bill fisheries, for enhancing the productivities. Then I told there are the self-recruiting species in terms of when going for the climate change. We practice the Gaducia chafra, then your Amblyphalidon mola, and you have Ompec pabda. So these are the wetland species, and we have found is a convincing and encouraging results has been given. And if you will find also some of the SIF is also used for the pen culture with the major cuts, and it was found very good growth rate also. So these are some of the growth rate of this. Then if you will see Manipur, again you have largest freshwater lake has been used for the grass carp and other pengwa models for finding the alternative good growths also. And this is also, and here total if you are finding when using the pen culture, when the productivity is increased maybe 1.8-fold to 3.3-fold, again the mandate has increased in the wetland fishing from 55 days to half an year. So it is interesting models where you can incorporate it into the wetland or small reservoirs, yielded larger benefit and community participations. Again, I told IMTA was practiced in a Bhembanar neck where the multi-trophic system is used in the pen, and we found there is a lot of benefits as done by the fishermen. Then we told we have developed some of these are the out of that. We have in the West Bengal, Bihar, Kerala and Assam, the climate resilient pen systems with the climate resilient species were developed. And also some of the things. So these are the inclusion people see when you are utilizing, even the captive nursery. People ask, why not captive nursery? But captive nursery is dried up certain periods, and the moment in the water does not happen in that way, small nursery. So the growth is less is compared to the pen culture. Pen culture is a higher growth rate compared to the captive nursery. And you can shift it. It is not only the fingerling productions. Some are used in the table size productions. It can be used for also selling of the fish, live fish also. So multiple aspect and multiple aspect that can be lead to be healthy productions, live fish transport, sell, and the livelihood, and it will have a net economic return is very high. And we can practice early time, and it's a low cost. Maybe 0.1 hectare is only Indian rupees of 60,000 rupees involvement. So that can be work for a FRP based, and that can be 10 years lifespan. So that is a beautiful systems developed by our institute, which are being operated across the country. Thank you. Thank you.
Thank you for your excellent presentation, Dr. Das. Any question?
Thank you. I have just one question. Thank you very much for your nice presentation. It's about the integrated multi-trophic aquaculture. Can you explain a little bit more? What kind of plants you are growing in the aquatic system, and you have shown the snails, and along with the fish, what are the uses of aquatic plants and snails? Can you explain a little bit more?
Here only one model we have tried, because if you'll see Kerala backwaters, the clams is very much eaten by the Keralians in that side, and Atroplos sudantensis is a high value species in that region. And we thought that how don't we use this in the bottom and the feed systems effectively, and we found it was successful. Again, that can be used in other part wetlands, because somewhere freshwater pole and other things people want to do, that can be utilized. Again, the plants, utilizing the plants and other things, because we have to analyze the wetland specific habitat vegetations, and other species may be benthic organisms. Mostly we are trying to incorporate with the benthic organisms, maybe your snails and other things which are of medicinal values and consumed by the lower strata of the people and the food species. And within that it will be, and because of pollutions, other things, the biodiversity, the molluscan biodiversity is a threat in many of the wetlands when you are going for the biodiversity. So if you look into all these aspects, again if you see when the benthic organism reach, maybe the bottom respirations and the bottom is very purest form. So we are trying to incorporate these are the practices, and because people do not understand initially first point of view why we should go in this aspect, only fish. So bringing into those people in a particular domain, it takes little time. So this is the approach where a holistic ecosystem modeling is required, then we can find out the suitability and go ahead with it. Thank you.
Thank you.
Thank you so much, Dr. Das.
Thank you, sir, for
Thank you, sir, for this nice presentation. Sir, I have seen your slide. You have done many experiments like polyculture, monoculture, and high-value fish culture also. Sir, you found the result. In which culture it is better? Which one is the best culture, either monoculture or polyculture? Number one question. And second question is, sir, can we do freshwater prawn culture in pen culture?
Yes. The last question, you can do freshwater prawn culture in polyculture. Then polyculture with SIF is the best option to utilize the whole aquatic bodies where you are encircled to be greater and better production.
Okay. Thank you, sir.
Let's give a round of applause to Dr. Das.
Thank you. Thank you.
With this, we come to the end of the oral session. Thank you so much to all the presenters for your well-researched and emphatic topics. Now let's go for the coffee break. There's a demonstration during the coffee break, and it's the processing of milkfish by Mrs. Sasiratikan Rungsasithorn, milkfish farmer community of Eastern Region, Thailand. Please feel free to engage in that. I have two announcements. Everyone is requested to come back to this room by 10:55 because we begin with the next session at 11 a.m. And the participants who have not yet submitted their presentations for the next technical sessions are requested to come to the booth here and do the necessary. Thank you so much.